important modules for the long non-coding RNA knowledge base is the lncBodyMap. Basically, this module is available through the lncBodyMap link. If you click on the lncBodyMap, it basically takes you to the body map, the different tissues that we've analyzed across the GTEx data. Basically, this shows a wireframe diagram of the body map and the various tissues that we've analyzed in the GTEx data are listed here. By mouse over on these tissues, you get the different metrics or different numbers of basically that related to each tissue. For example, how many whole genome sequence samples in the GTEx data that we had available to perform our analysis, how many SNPs in the EQTL analysis that were involved, how many genes for each tissue, how many protein coding genes versus long non-coding RNAs, and so on and so forth. So if you mouse over each tissue, you get these metrics, but an interesting feature of the very interesting feature of the lncRNA-KB is that if you click on any of these tissues, so if we take an example of the brain, it takes you to the tissue-specific page and different features of data that are available for each individual tissue, and you can go back and forth between the body map, or you can select any particular tissue that you're interested in to get these data that are available for each individual tissue. So for example, going back to the brain, we have 4 different sections for each individual tissue. The first one is the gene expression data that is available for each individual tissue across each tissue. The gene expression data is basically from the GTEx project data. We make available the raw count gene expression matrices across the lncRNA-KB, the TPM normalized feature counts, gene level and transcript level matrices, quantified by Salmon, as well as the TPM for gene level that is quantified by Salmon. And we also make available the statistics, how many reads were assigned by sample across the particular tissue. In addition to the gene expression data, we also make available the EQTL data for that tissue. In the EQTL data, we have the Manhattan plot, which is the genome-wide EQTL results, as well as the whole file of results, basically across all the SNP gene pairs that are defined by the certain criteria that we've specified here. Basically, this is a genome-wide EQTL results across the lncRNA-KB involving all the long non-coding RNAs and protein coding genes. One of the main components of the tissue-specific page or the body map is the network and enrichment pathways. This is basically what we did was we tried to potentially characterize or functionally characterize the long non-coding RNA using the weighted gene co-expression network analysis. So we performed a WGCNA analysis for each tissue, and all the results for every tissue we make them publicly available across the long non-coding RNAs and protein coding genes. So here, when you go to a particular tissue's networks and pathway enrichment page, you will see a graphical summary of the WGCNA modules that were identified in that tissue. These are the interesting pathways, interesting processes for that tissue for which those modules are involved in. So here you can see the involvement of specific modules in synaptic transmission or RNA splicing reactions or forebrain development. So these are all relevant to the brain tissue, and we just wanted to give an overview of the WGCNA results using this network enrichment map. Then if you go further down, we provide all the significant modules and pathways, basically results for that tissue. Basically here we have the whole all the modules that have been identified in that tissue, and the pathways that are Q-value significant. You can download the entire list of pathways that were Q-value significant across the different modules, and you can actually sort by Q-value, you can sort by P-value, you can actually search for different keywords. For example, brain, you can search for any keywords that you prefer, for example, brain related processes, and hopefully that will give you the information on modules that were identified using the WGCNA. And another portion of this is that we actually selected 25 notable pathways based on some curation and knowledge. We selected 25 notable pathways that were related to the brain across various modules. And for each of these notable pathways, we provide the dynamic network generated using Cytoscape, and users can actually view this dynamic network accordingly and click on any gene which will actually focus in on that particular node. You can search for that gene linked to gene cards or NCBI, and you can go back and forth on different networks. You can search for long non-coding RNAs, basically the LNC IDs or any other IDs based on these networks, and download them as well. So you can really understand the involvement of long non-coding RNAs in these processes. We also give actually you can get the full list of modules across the brain. You can also download the adjacency matrices across the brain, and this is available for every tissue. You can download it by module as well by just clicking down on this by using this drop-down menu, and for each module you can then download the adjacency matrix, which can then be exported into R or Cytoscape for visualization. And then the last part of the tissue page or the body map is the trait association. So we give the results of the brain tissue across the 323 GWASs that we ran or that we tested the association of trait, and prioritized for each trait. We prioritized the genes that might be involved in any of these traits of the UK Biobank. And as you can see here, these are the top table actually shows all the traits, the 323 traits that we analyzed the brain tissue against, that we tested the brain tissue for. And in the bottom here, by clicking on any phenotype, you will actually it will take you to the UK Biobank information page, which shows the different heritability estimate for that particular trait, as well as the number of cases and controls, and some more information. In terms of the results for each trait, we provide the actual summary Mendelian randomization results, which are p-value SMR less than 0.05. So from this table we can see that in this trait these genes were prioritized in the brain tissue, and users can actually sort the results by any of these criteria that they might be interested in, particularly the PSMR, and using that they can then go and focus in on any particular gene, and this links back to the gene page, or this links to the SNP information in dbSNP, and you can go back and forth between the tissue page and gene page to see if there's any gene of interest for that particular trait. And by clicking on the particular trait, the table dynamically changes to that particular trait, and you can go through each and every trait, um, based on any keyword that you would like to search. So if you search for depression, for example, which is relevant to the brain, you can see that they have a um, non-cancer illness code, self-reported depression, in the UK Biobank, which can be used, which across which we have ran the SMR results for the brain tissue, and you can see that these are the results of the SMR, which can then be sorted by p-value, ascending or descending, and these are all SMR less than 0.05, and then users can actually link back to the gene page, which you get all the information related to that gene, and check the expression across different tissues and see if this gene was involved in any of these brain-related processes, what the tissue specificity of that gene is, the EQTL results across the different tissues, and that really gives you the user an advantage of looking at tissue-specific results across the GTEx data for long non-coding RNA.